All right, well, thank you, everyone. I appreciate Noble giving me the opportunity to conclude today's sessions. I will aim to make this one very memorable for you. Speaking of memorable, I know all of us in this room are old enough to remember, let's say, 20 years ago, when there was a lot of excitement about something called cell therapy. And it promised all sorts of wonderful and amazing things, new medicines, et cetera, et cetera. There were many scientists that were touting this new era. I was one of them, in fact. And I've got good news and bad news. The bad news, of course, is that 20 years have gone by, and with the exception of cancer, cell therapy hasn't really produced a lot of the things that it was promising to produce. However, the good news is it's finally starting to happen. And you can measure this in certain ways. You can measure this in the development of products, but you can also measure it on the capital side. And because this is an investment conference, you can point to things like the $1 billion acquisition of Blue Rock Therapeutics that works in this space, or the $950 million acquisition of Semma. Or now, more recently, you can also include a deal we recently struck with Roche for a license for our lead program that has a value of up to $670 million. So there are things happening in this space. It's a maturing field, and it's really exciting. And I'm looking forward to telling you about what Lineage does in this space. So first, I refer you first to our safe harbor clause. I may be making some forward-looking statements throughout the balance of this presentation. So Lineage and some of those other companies I mentioned, we are advancing a new branch of medicine. It is the use of whole cells as the medicine. So rather than small molecules or antibodies, the things that we're usually familiar with in terms of therapeutics, we're actually using an entire cell to treat the patient. This is a summary slide of our technology. It's based on manufacturing and transplanting these whole cells. We have these validating partnerships. I mentioned the one with Roche and Genentech. We also work with CERM and Cancer Research UK. The exciting thing about using a whole cell as medicine is that sometimes the entire cell can do more than just a small molecule, especially in areas where small molecules have failed. And I'm going to show you some examples of that. I'm going to skip to something that I always get asked about. The company's cash position at the last reported quarter was about $83 million. And to give you some perspective, the last couple of years, we only spent about maybe $25 million. So we think we have abundant runway to be able to reach some important milestones in the programs that I will talk to you about today. This is a snapshot of our clinical and non-clinical programs. There are 4 of them. Oprogen is to treat dry age-related macular degeneration. We have a spinal cord injury program. We have a cancer program and a brand new program internally generated for auditory neurons. Now, what these programs have in common is they all are based on the manufacture of a specific cell type from a stem cell starting material. So I want to be really clear and kind of restate that. We never put a stem cell into a human being, but stem cells are very powerful, and they can become the starting material for other cell types. So for example, if you have a disease which is characterized by the loss or death of retina cells, you could use stem cells, and from those stem cells manufacture retina cells and replace the ones that are lost. And that's been the basis for this large deal that we did with Roche. And we have similar approaches in the spinal cord, where we manufacture spinal cord cells, in oncology, where we manufacture part of the immune cells, and now also auditory neurons to address hearing loss. These are the 3 steps. It's really simple. You start with these pluripotent stem cells. Those cells are capable of unlimited expansion. And they don't change. This is the real power of this underlying technology. Those cells can be increased in number. Many, many billions of cells can be manufactured. Then in step 2, you convert them. And it's almost like a recipe. You convert them through natural processes into the cell type which you desire to have. I called it like a recipe. And just like flour can lead to a cookie, or flour can lead to bread, and you know that cookies and bread do different things. It's the same with our starting material. These pluripotent stem cells can become retina cells, liver cells, kidney cells, heart, brain, bone, blood, anything you can imagine. Of the 200 cell types in your body, these pluripotent stem cells can become all of them. Thirdly, we then develop the product. So after we've manufactured the specific cell type and tied it to whatever disease or condition that we're looking to treat, we then use those cells, we transplant them to the patient, and if they are functional and they are integrated and they're not rejected, which has been our data to date, then you may be able to see some clinical benefits. The amazing thing, which I sort of referred to, is because you have 200 cell types in your body, in theory, we could have 200 or more different programs. The ones that are highlighted in orange are the ones that we have today. But again, this is a platform which has tremendous breadth and power, and we could utilize this platform to go into many different areas. Most of our experience has been what's called the ectodermal or neuroectodermal lineage. So that's at the bottom. That's where we're manufacturing the auditory neurons and the specialized retina cells. But we could expand. We could go into other areas. But these are our areas of focus for today. We're trying to go after areas where there's a compelling unmet need, large market opportunity, and a paucity of competition. We do all of this internally. We do not throw our manufacturing over the wall. If you're making a small molecule, sure, you can get a contract organization, they can make the molecule, you give them the picture of it, they'll go make it for you. With whole cells, it's much more difficult, many millions of times more complicated because you have many millions of molecules that comprise the cell. So it's very difficult, and it's really important to retain your own manufacturing, control your process, and of course you can generate a lot of intellectual property. And in fact, we do have one of the largest intellectual property estates in cell therapy with more than 600 different applications and pending applications. I'm going to briefly go through what we're doing in the setting of dry age-related macular degeneration. This is one of the leading causes of blindness, and this is the basis for this worldwide collaboration we have with Roche and Genentech. This came with $50 million up front and another $620 million of additional payments, and we announced this in December of last year. So this is still fresh. We're still launching this. Genentech is going to take over all of the development of the product and ultimately commercialization of the product. We are doing some manufacturing still for them, and there are joint steering committees that oversee this work, and we are completing the ongoing study. The data that we are already generating will continue to follow up those patients. Why did Genentech and Roche, why did they want to do a deal? Well, there's a tremendous unmet need here. In the setting of dry age-related macular degeneration, the loss of retina cells leads to an ever-growing area of atrophy shown by these 6 panels. As you go from left to right, that area of atrophy is characterized by the death of retina cells, and it just goes outward. I'm from Southern California. We have forest fires out there. This is just like a forest fire. It just expands outward, just consuming and destroying everything in its path. What it's destroying is your vision. This person started with 20/20 vision, and as you can see off to the right-hand side, in just a few years, they have become legally blind and really have negligible vision after 7 years. So what can we do about this? Well, first, the commercial opportunity is quite compelling because unlike the other form of AMD, which is called wet, for which there are $10 billion of sales, this dry form of AMD has no approved products and is 8 to 9 times more common. So we have a tremendous unmet need, no FDA-approved therapeutics, and probably a very significant commercial opportunity. Now, people have tried to develop drugs in this space and failed. We are trying a different approach. We are, as I described, manufacturing replacement cells. So on the left side, this cartoonish figure, the schematic, this is showing the death of cells. Those cells are in the retinal space. They support photoreceptors above them. When you lose those RPE cells, those are the cells we make, the photoreceptors lose their nourishment, they fail to function, you lose your vision. So if you can replace them, on the right, those shiny blue cells, those are the replacement cells. So conceptually, this is kind of like a bone marrow transplant, right? Make the cells, put them in the patient who is losing the cells, look for benefits. We have been tremendously successful on the manufacturing side. This is actually sort of a hidden strength of the business is our manufacturing capability. We are manufacturing essentially pure populations of RPE cells. You cannot detect residual undifferentiated cells in our clinical material. So everything is pushed into becoming a retina cell. We never put a stem cell in a human being. We always convert them over to some other cell type, the cell type which is deficient in the body. We've developed a new thaw and inject formulation, which makes it really easy to use this stuff. You take it out of a frozen state, 5 minutes in a warm water bath, into the syringe, 30-minute surgery, and the patient is out of the surgical suite and is able to go on with their life. So it's a straightforward clinical procedure to administer the cells into the subretinal space. By the way, these patients are familiar with having injections into the eye. I know it sounds horrible, but hopefully none of us will get there. But if you're going to lose your vision and you're faced with having an injection in your eye, it's actually a pretty easy decision. We have been very successful at increasing the manufacturing scale. I definitely challenge you, if you look at other cell therapy companies or regenerative medicine companies, really ask tough questions about manufacturing. We're at the point that in a 3-liter bioreactor, we can manufacture about 2,500 clinical courses. And this 3-dimensional growth, this is key, growing in 3-dimensional space instead of flat plastic plates, gives us tremendous efficiencies and also tremendous control over our process. And you can run larger bioreactors, you can put them in series or in parallel. So it's important to have a line of sight into commercial production, and we have that. The most compelling thing about what we have done is we've shown data that no other academic or corporate group has shown, which is we've been able to show that an area of atrophy, similar to those pictures I showed you a moment ago, can actually become smaller. Areas of retinal loss can become restored after transplants that we provide. The reason why that's a big deal is because it never happens naturally, right? You could rip an arm off of a starfish and it might be able to grow it back, but you cannot remove retinal tissue or lose retinal tissue and regrow it. That's not a function that human beings can do. So if you take a picture of someone who has lost retinal tissue and then you treat them and you come back and you take another picture and there's new retinal tissue there, it's because of our intervention. That's really exciting, and this is sort of flying in the face of conventional wisdom for this disease condition, because heretofore, almost all of the leading companies are trying to slow the process down. They're trying to have that forest fire expand slower. That's best attempts right now. We've been able to not only stop it in some patients, but even reverse it in some patients. So I think that is probably the most compelling data that led to, as I say, this up to $670 million deal with Roche and Genentech, which to our knowledge is, outside of oncology, the largest cell therapy license agreement that's been signed. This feeds into this whole proof of principle that we've been working on. I joined the company about 3.5 years ago. Everything that we've done here since I joined is really about demonstrating that the use of whole cells in therapeutic settings may be able to generate outsized clinical benefits as opposed to some of the other traditional approaches that have been pursued. So we're going to keep going with it, right? The story is not over, certainly, with a big pharma deal. We're using some of that capital to reinvest into some of our other programs. We are manufacturing a special kind of cell in order to help patients who suffer from spinal cord injury regain mobility. This is a young man who obviously crashed his car. He was on our clinical trials. He used to be quadriplegic. He could barely shrug his shoulders, and this is him throwing a baseball. What's really actually more important is the water bottle, right? What's more important is that this kid can take a drink of water when he wants it, or he can manipulate his wheelchair when he wants it, right? That's wrist control, right? Bicep control. That is what we want to do. Everybody has a cell phone. You want to be able to use your cell phone, right? Upper extremity mobility is where we're focused. And I know 20 years ago, people said cell therapy, we're going to get people getting out of their wheelchairs. Yeah, maybe someday we are. But let's at least start somewhere. Let's start with a little bit of upper extremity mobility. Wouldn't that be a good thing? We believe that it is. This is actually a fairly common event, maybe 18,000 times per year. It's what you would expect. It's accidents, car crashes, and it's very sad because oftentimes it is young men who definitely did not expect this to happen. You smoke 5 packs of cigarettes, you might get lung cancer, but nobody expects to become immobilized from a spinal cord injury. I've really covered the unmet need, and I've covered the point that we want to gain mobility into the upper extremities. I want to show you how we're going to do this. We manufacture oligodendrocyte progenitor cells. These are the cells which are responsible for creating the myelin insulation around axons. It's a technical phrase, but suffice it to say that in order to have Cohesive mobility, to organized mobility, not spastic mobility. You have to have nerves that are all wiring together and all firing together, and those nerves need to have myelin. And so we're producing and transplanting to these patients the cells that are necessary to myelinate the axons that they rely on for their mobility. They get, just like in the setting of dry AMD in the blindness case, these cells are injected directly into the spinal cord. So the approach is very similar to the success we've had with Oprogen. RMAT designation, orphan drug designation, $14 million from CIRM. There are a lot of nice attributes to this program. And similar to the dry AMD program that I described, there have been 25, actually 30 total patients treated in this. In the dry AMD program that I described, we had treated 24 to 25 patients with various ophthalmological disorders, levels of distress. We've got 5 patients with thoracic injury, which is a little lower, and then 25 patients with cervical injury. So in terms of clinical data, clinical evidence, it's actually pretty comparable to what we have shown with the dry AMD program. This is what we're doing. You can see on the left, this is a control. This is an animal spinal cord. This is just to demonstrate what we're doing inside the patient. I'll show you an MRI, what it looks like in the patient in a minute. But the concept here is that gap, that vacancy, it's called a cavity, that hole, electrical impulses cannot jump across, right? Just like a cord in your house. You can't jump across the air. So you have to have some material, you have to have some substance in there. So we manufacture those cells, we transplant them into that cavity, and when they're there, they're manufacturing myelin, they're secreting neurotrophic factors, they're giving the patient something to work from. Physical therapy, electrostim, great, bring it on, add it, we want it, because we're giving you the material that can respond to that additional therapy. These cells are coming from an NIH-registered cell line. We treat patients 3 to 6 weeks after injury. I make that point because if you've got 3 to 6 weeks before you treat a patient, you have plenty of time to relocate them if you need to, if they're not at a level 1 trauma center or wherever. If you need to get, and you do, all of the appropriate permissions and approvals to conduct this sort of work. This is not like the setting of stroke, where you often hear about the golden hour. You have to be, literally there at the diving board where the kid went into the pool. We have time to get the patient ready and select the right kind of patient for this kind of therapy. This is some animal data. It's just to show you that when you do this in animals, you can regain additional motor function. And this is a different version of the same thing. I'm not going to try and make this a data-heavy presentation. I'm also not going to go into the subsets of all the different kinds of patients. All of that's available. I'd love to talk about it another time. What I do want to stop on, though, is this. This is the safety output from the 25 patient trials. What's really compelling here is that you have more than 500 adverse events, only one of which was potentially related to our cells. So I think this is really compelling because it says that the safety profile, the tolerance of this therapy is very favorable. So what that means to me as someone who runs clinical trials is that if we show even just a little bit of benefit in these patients, I think the risk-reward calculus is compelling and in our favor. Most of the patients, I'm going to say 80% of them, do develop that cavitation. Only one of them in our clinical trial. So we reduced cavitation from 80% to 4%. And that cavitation is associated with different complications and other adverse events. So really happy to see that the cells have been retained, they've been stable, they're not rejected. These patients are not on lifetime immunosuppression. Like a lot of the stuff that you maybe heard about or were afraid about or thought skeptically about in terms of cell therapy has not shown up, at least in our hands, in our clinical trials. So I think that's encouraging. More encouraging than that is this. 96% of patients in this setting had some improvement in their motor activity, and approximately a third of them, had 2 or more levels of benefit. I don't have time in this presentation to go into what that means. There's a scale that looks something like this, but I'm going to skip over and I'm going to give you something that you can relate to maybe a little more easily. If you gain 2 levels, this is essentially like pushing the injury down lower and gaining function higher. You can see what it means to someone to move from C4 to C6 or C5 to C7. Massive reductions in cost of care, huge gains in quality of life, right? Ability to go to the bathroom when you feel like going to the bathroom, ability to button a button on your shirt. These are important gains for individuals who fit into this profile of cervical level C4 to C7. We also think we can do better. We noticed some things in the data that we think we can avoid next time. The two worst patients, by the way, had this cord compression. You can put a port in the patient, relieve the pressure, and we may be able to select for better patients in the next study and get better outcomes. As part of a future design, this is something that we're not starting until next year, but it's an important consideration for how we're learning through this new technology, how it can be better. I'm going to skip the cord compression. I'm going to skip some of the specific data. A lot of this has been or is currently being published. We are, as a next step, near term, going through some improvements in this program. The original way of administering the cells requires this big scaffold and everything. Now we have this cute little device. We're going to start this clinical trial later this year in 5 to 10 patients and demonstrate that it is an effective way of delivering the cells. The cells themselves have been vastly improved in our hands. We have this new ready-to-inject formulation, scale, all these manufacturing things that we learned from the ophthalmology program, we're now applying to the spinal cord program. This is an example, the purity of our cells. This is a table showing you levels of impurity. Blue is the original stuff that was used in the clinic. Orange is the stuff we're making today. A lot cleaner, a lot more pure. I think that's going to be beneficial, and I think the FDA is going to be happy about that. So I'm going to keep going through because I know I'm going to run out of time here, and I want to tell you about two other similar programs. One is that we can manufacture a third kind of cell, a dendritic cell. Dendritic cells are part of your immune system. They are very good at delivering information to your immune system. They can carry, that's their job, that's their natural job, is to carry antigens to your immune system. Because we can manufacture dendritic cells, we can load those dendritic cells with 50, 60 million copies of an antigen and put that in a patient. So the patient's immune system is getting a screaming loud education as to what foreign material looks like. So instead of replacing cells that are absent, in this case we are just adding cells that have a very specific function, right? So it's a little bit of a spin on the other examples of cell therapy. But it's really exciting because you can go in a lot of different directions because it's not the cell that's therapeutic, it's the information that makes it a therapeutic. So if you want to put a colorectal cancer antigen into this cell, you can. You want a breast cancer antigen into this cell, you can. There's a lot of different partnership ideas that we have here. I have to skip because I really got to tell you about this new one. It's hearing loss. 430 million people around the world suffer from hearing loss. Fundamentally, we have a problem where as you age or you hear a lot of, you went to too many rock concerts or some medications can do this, you can lose your auditory neurons. Let's make the auditory neurons, let's replace the auditory neurons. So this is a new program. We didn't acquire this. This is internally built by us. And if you go back to that first slide I showed of all the branching of all the things that you can do, this is an example of it. This is something that 6 months ago didn't really exist. And we've been able to create this new program, and now we're launching this new program. We're working to get this new program into the clinic. It's the power and I think the potential and perhaps the great value that Lineage can offer to investors is being able to expand the scope of work that we have. I'm going to go through, we've got a bunch of clinical and regulatory milestones. I want to just do the takeaway to say Lineage is a company that I believe is a leader in this regenerative medicine / anti-aging space. We are a cell therapy company, but again, we don't put stem cells in people. We are more like a bone marrow transplant. We use a specific cell as the medicine for the patient. Company is well capitalized. We control our manufacturing. We control our own fate in a lot of ways. And I think what we have been able to do for patients, which you can find examples of on our website, have been very compelling in terms of their ability to move, their improvements in vision, and some of the credibility of this new corporate partnership, I think puts us on a wonderful trajectory. And I certainly look forward to the overall healthcare environment and biotech environment starting to reward companies for the kinds of things that we are doing. So thank you so much for your attention, and I will conclude there.